Signal propagation is the process of sending information from neurotransmitters to the brain through different and complex processes, such as the packets of information sent through the action potentials, the neurotransmitters that are released through the axon, and the synapse that helps to transfer between two cells or target cells. The central nervous system is composed of two kinds of cells: neurons, which are about 10 to 11 number of cells, and glia, which helps with every information processing and controls our mind-related functions. The action potentials help to transfer signals from neuron to neuron when the axon membrane rapidly depolarizes and repolarizes. Action potentials are also called electrical impulses that send signals throughout the body caused by the continuous motion of ions exiting and entering the cell to maintain a negatively charged cell. Almost every neuron has a single axon, which is extremely essential for the conduction of action potential away from the cell body and towards the axon terminus. When the action potential is at its highest, potassium channels open and begins to leave the cell, and at the same time, sodium channels close, which leads to hyperpolarization of the target cell. When the neuron is in a resting state, which means that a neuron has more sodium ions outside the neuron than more potassium ions inside, which means that it's more negatively charged than the outside of the neuron. Synapses are also called neuronal junctions, which are the site of transmission of electric nerve impulses between two nerve cells. A typical synaptic cleft, which is the narrow space between two cells, would be about 0.02 micron wide, which allows the neurotransmitters released from a cell to diffuse across the cleft by taking about 0.5 milliseconds or so for them to stick to receptors on postsynaptic cells. The two parts of the synapse is presynaptic, which is before the synapse gap, and postsynaptic, which is after the synapse gap, which is most likely near the target cell. The process of binding the neurotransmitter changes the ion permeability of the postsynaptic cell membrane. The synaptic fluid is filled with extracellular fluid and creates a physical barrier for the electric signal to be carried out by a neuron to another.